Hello. Today's online lecture will be for you to study during the Thanksgiving. We will talk about urinary system. The first thing that comes up with your mind when you talk about urinary system, most of the students will probably come up with kidneys. And yes, today we'll focus on the kidneys. But before that, let's talk about the general functions of the urinary system. So, the general function, of course, is to excretion of the metabolic waste, and all these are nitrogenous waste. Urea is the primary one, it's also the most famous one. Many of you will have heard about this already. Urea is basically when your liver break down the excess amino acid that you have in your body, then the liver will convert the excess amino acid into urea. And then we also have ammonium, creatine. Creatinine is from creatine phosphate. Creatine phosphate is like the immediate short-term fuel, short-term energy for your muscle contraction. And then uric acid. Uric acid is from nucleotide. Osmoregulation. Kidney or urinary system is for regulating the amount of water in our body. It is important because if we have, the more water we retain in our body, the more blood volume we have, and when we have more blood volume, then we have higher blood pressure. So by regulating the amount of water in our body, we can adjust our blood pressure. And how do we adjust the amount of water that we absorb into the body? This is the key thing that you have to memorize in order to understand the rest of the lecture, is that we regulate the absorption of salt by regulating the amount of salt that we absorb in the kidney back to our blood. Then we can control the amount of water that we absorb, because water always goes with the salt by osmosis. Kidney also reabsorb other ions like potassium, bicarbonate ion, and calcium. Bicarbonate ion is very important ion to regulate the blood pH, because bicarbonate ion is like a buffer that helps the blood to maintain pH 7.4. And we will talk about the reaction of bicarbonate ion at the end of the slides, at the end of this lecture. We have the acid-base balance. In here, just like the bicarbonate ion, the pH of the blood, they have to be in 7.4. And not only the urinary system, but the respiratory system, that means our lung, also help to regulate the blood pH. At the end of today's lecture, we'll also talk about that, briefly talk about how our lung regulate the pH. And our urinary system can excrete the acidic hydrogen ion and reabsorb the bicarbonate ion, so that the bicarbonate ion can maintain our blood pH. And then it also helps the secretion of hormones. Our kidney produces three things. Two of them are hormones. One is called erythropoietin and vitamin D. Yes, vitamin D we consider as a hormone. But kidney also secrete renin, but renin is not a hormone. I added this part to the slides. Renin is not a hormone. Renin is an enzyme, not a hormone. Secretion of renin will induce the secretion of aldosterone. Aldosterone is will induce the absorption of the sodium. Sodium, as I told you in the previously, absorption of the salt will help to reabsorption of the water, retain the absorption of water. So, aldosterone will decrease the urine volume. Erythropoietin is a hormone that can stimulate the manufacturing of the red blood cell from our bone marrow. Vitamin D, well, yes. We all know that if we have a suntan, then the skin will make vitamin D. But kidney actually will so-called activate vitamin D. That is, and then you will have to promote the absorption of calcium. Kidney, they are bean-shaped, reddish-brown organ. It is behind the peritoneum. And not only that, it is behind our digestive system. So in Bio 3B lab, at the end of this semester, you will have a pig dissection, and then you will have to remove all the digestive system, the gut, the stomach, the esophagus, etc. And after you remove all of them, you'll see a membrane covering it. Then you remove that membrane, then you will see the urinary system. Mainly, you will see the two kidneys. So the digestive system is ventral, while the urinary system is relatively dorsal. And then it is covered by something called renal capsule. Renal capsule is basically a tough membrane, tough fibrous connectivity, a tough membrane that wrap around the kidneys. It is different from the pericardium, the membrane that wrap around your heart, or the pleura membrane. The pleura is the membrane that wrap around your lung. It is different. It is a tough fibrous connectivity mainly to protect the kidneys. And then on the kidney, we have something called hilum. Hilum is a depression that allows the renal artery to go in and allow the renal vein and ureter to go out. Ureter. Basically the major function of ureter is to connect the kidneys to the bladder. So this is the urinary bladder. So this is the major function of kidneys. And they transport the urine by peristalsis. Remember this term peristalsis from the digestive system. Yes, our ureter also use peristalsis to move the urine. And also remember, in digestive system we have 4 layers of different tissues, 4 layers of different cell type tissues in the digestive tract. In here, ureter only have 3 different layers: mucosa, smooth muscle, and connective tissues. Urinary bladder. The major function is to store the urine, and then the urinary bladder is stretchable because they have a circular fiber and 2 layers of longitudinal fiber. And there's something called transitional epithelium. We did not cover transitional epithelium in the Human Organization Online lecture, but you will learn about this when you take anatomy. Basically, transitional epithelium is that when the urinary bladder is empty, that means the transitional epithelium looks like cuboidal cell, but when the urinary bladder is full, then the urinary bladder will be stretched. When it is stretched, then the transitional epithelium looks like a squamous cell. We call it transitional because this epithelium cell type, they change the shape from cuboidal to to squamous when it is full. When after we pee, after we urinate, then the bladder shrink and the cell go back like a cuboidal shape. Bladder has 2 sphincters. Sphincters are the muscle that can constrict or relax. When it constricts, it stops the urination. When it relaxes, then urination can occur. Urethra. Urethra, they are the exit of the urinary bladder. It is longer in male than female. It is important, this one, because of this reason, female only has a very relatively short urethra. Because of that, female get bladder infection easily. Because bacteria only need to travel a relatively shorter distance, the shorter urethra, to get to the bladder in the female. But male urethra is a lot longer, so bacteria need to travel longer distance. By the time they crawl their way up, then we already pee again. So peeing or urination actually helps to clean up the bacteria as well. And then we also use the same urethra for semen. We'll talk about that at the end of the last lecture. We will talk about the reproductive system. So this is a basic schematic diagram of the anatomical position. So kidney produces urine. In today's lecture, we will focus mainly on this part, how kidney produces urine. The urethra transport urine to the urinary bladder. Urinary bladder will store the urine. Urinary bladder can store approximately 250 milliliter of the urine. And then urethra open, and then we can pee. And I want to point out this diagram here. I would say all the textbooks, they will draw that the right bladder, the right kidney is higher than the left kidney. And it is not a mistake, it is anatomically correct. Can you come up with a reason why the left kidney is lower than the right kidney? The main reason is that there is a gigantic liver on the left side of the body that will press down the left kidney. And it is natural and it is healthy. So don't worry about, oh, I have an asymmetric kidney. No, everyone has an asymmetric kidney because the left side here we have a bladder on top of the kidney. So urination, another name for that is micturition. Urination or micturition usually is 250 milliliter, 250 milliliter is basically the amount of coffee you can put on your coffee mug. The ceramic coffee mug is approximately 250 milliliter. So let's say we always heard that we need to drink what, 8 cups of water. 8 cups of water, each cup is 250 milliliter. That means it's approximately 2 liters. So they tell advertisement or mass media tells us that, okay, you guys, it's healthy to drink 8 cups of water, meaning that 2 liters of water. You know why? We'll tell you why later. Because each day we basically urinate or get rid of 1.8 milliliter of urine. You can see it in the next few slides. So when urinary bladder is full, then the stretch receptor on the urinary bladder will send a signal to the spinal cord, and then it will tell us to relax a sphincter. When the sphincter muscle relax, then we can urinate. We were potty trained by our parents so that we can control our urination. So this is the schematic diagram to show you the connection of the nervous system. So the kidney has 3 regions. So of course the outermost part, the kidney is wrapped by the capsule, renal capsule, which is the tough membrane. But the actual kidney, the surface part of the kidney is called renal cortex. And inside of the kidney we have the renal medulla. Renal medulla, they are the cone-shaped tissue, we call it renal pyramid. Inside the cone-shaped renal pyramid we have nephron. Nephron, they are the subunit, basic subunit of kidney that produces urine. We will spend most of the time in today's lecture to learn how nephron produces urine. And then we have renal pelvis. Renal pelvis is basically a gigantic funnel. A funnel that connects to all these renal pyramid, and all the nephron, they will produce the urine and being collected by the renal pelvis, and the renal pelvis will connect to the ureter. This is the basic structure here. All these cone shape, they are the renal pyramid. So the cortex is here, renal cortex, and then the renal medulla. They are basically all these renal pyramid here. This is the renal pelvis here. So you can see that the nephrons are here. We have a bigger picture for nephron later to show you. And yeah, it shows it here. The renal pyramid, renal cortex, and renal pelvis. And then the urethra is basically the extension of the renal pelvis here. Now, the nephron. We have millions of nephrons. That's why the kidney can filter, or they can filter approximately 180 liters of water every day. And as own blood supply, so the major part is called glomerulus. Glomerulus, they are a network of capillary. The input of this glomerulus, or the network of capillary, is called afferent arterial. Afferent is the input to the glomerulus, the network of capillary. The output of the glomerulus is the efferent arterial. So remember, A is the input, E is the exit. So E is the exit, E and exit, they start with E, efferent and exit. And then the efferent arterial will take the blood to the peritubular capillary capillary network. Capillary network. This peritubular capillary network, they are the blood vessels that surround the nephron. The nephron, they have a different tubule we'll talk about later. And then after they surround the nephron by this peritubular capillary network, then they will become the renal vein to go out of the kidney. So in other words, all the afferent, efferent, peritubular capillary network and the renal vein, they're essentially the same blood vessels, but it's just that they go through different parts. So here is the schematic diagram here. You see that this is the afferent arterial that goes into this network of capillary. We call it network of capillary. We call it glomerulus. The network of capillary we call it glomerulus, and then the exit we call it efferent. And then one thing that I want to point out is that if you look at this picture carefully, the afferent arterial is actually a little bit bigger than the efferent arterial. There's an important reason for that. The bigger blood vessels here and the smaller blood vessels here is to create a high blood pressure in the glomerulus, so that the high blood pressure inside the glomerulus here will squeeze, or you can call it squeeze, or it will filter the solution or all the substances into this Bowman capsule here. The Bowman capsule, also known as glomerular capsule, it is the beginning of the nephron. So that it will go through all this nephron tube, and then eventually you will go to collecting tube, and then go to the urethra. And then we have the peritubular capillary. So this here is the peritubular capillary, and peritubular capillary. So the peritubular capillary will surround the different part of the nephron tube. We'll talk about later. But you just need to know, right now you need to know, okay, glomerulus, they are a network of capillary. The input is big, the afferent arterial is big, the efferent arterial is smaller, so that it creates a positive pressure inside this glomerulus. And the positive pressure here will push the substances out from the capillary into the Bowman capsule. The Bowman capsule is the starting point of the nephron. We call this filtration, or we might actually call it a glomerular filtration. We'll learn about this later. glomerular capsule, Bowman capsule, they are cup-like structure that's completely surround the glomerulus. If this is glomerulus, then the Bowman capsule, they are like this. It's like your baseball glove holding your baseball. It's like a baseball glove holding a baseball, completely surrounded. And then inside we have something called podocyte. Podocyte, they're specialized cell that they actually stick on the glomerulus. What they do is that they form pores of small size. The pores they form by sticking those podocytes on the surface of the capillary of the glomerulus. It regulates what kind of substances can go through, what kind of small molecule can go through. Only very small molecule can go through the pore because they're limited by the podocyte. Well, how does that work? The pore is so small, only like small molecule like glucose, urea, or water can go through. But large molecule like protein or red blood cell, white blood cell, they cannot go through of this small pore. And then the first kind of tubule that, we have the peritubular capillary, right? Peri means surrounding, tubular, surrounding tube. What kind of tube those capillaries surround? The first one is called proximal convoluted tubule, PCT. Proximal convoluted tubule, they are formed by cuboidal cell with microvilli. Remember, microvilli, they are the extension of the cell membrane. When the cell membrane extends to form microvilli, it will increase the surface area and for absorption. So proximal convoluted tubule, they are here. And then in this part we have the peritubular capillary here. And then we have something called loop of Henle. Loop of Henle is the U-shaped nephron tube. It is lined with a simple squamous epithelium. That means it is very thin. A lot of reabsorption occur in the loop of Henle. I will explain to you why in the next slide. And then we have proximal, of course we have distal. Distal convoluted tubule, DCT. They are also formed by cuboidal cell, but they do not have a microvilli. They have lots of mitochondria, but they don't have microvilli. When you see the part that have a lot of mitochondria, then you can assume an active transport is there. These are tubular excretion rather than reabsorption, so it's mainly for excretion, because it is the end of the microtube, it is the end of the nephron. And then we have collecting duct. Collecting duct is basically a major duct that connects with different microtubule. It connects with different nephrons. So a lot of nephrons, they will join together to form these collecting ducts, and then collecting ducts will go to the renal pelvis. Yeah. So we can take a look at this here. The loop of Henle is here. So after the proximal convoluted tubule, it becomes the loop of Henle here. Loop of Henle here, you can first of all you will see that it is big in here but small in here. Can you think about reason why? The reason is exactly the same as the reason here. The afferent artery is big, but the efferent artery is small, so that it creates a positive pressure in the glomerulus. Similarly, big at the beginning and then small here. You will create a positive pressure inside the loop of Henle here, so that it will facilitate the so-called filtration, meaning that the positive pressure here will squeeze the filtrate out, so that the capillary network here will be easily to reabsorb. So the structure here of the loop of Henle here is basically to create positive pressure to facilitate reabsorption. And the peritubular capillary network here has a special name. It is called is called vasa recta. Vasa means blood vessels. Recta means straight. As you can see that we have one straight blood vessel and two straight blood vessels here. The straight blood vessels here and also the network of the blood vessel, collectively we call it a vasa recta. And then we also talk about the distal convoluted tubule here. We call it proximal. Proximal meaning that it's closer. Distal meaning that it is farther away. Why it is proximal convoluted tubule is closer to the Bowman capsule. Distal convoluted tubule here is farther away from the Bowman capsule. And then we also have a peritubular capillary network here. And then connecting duct, you see that all the branches here, they connect to a different nephron. So this is a picture from electron microscopy. You can see that the arterial, the afferent arterial here, is actually a little bit bigger than the efferent arterial here. And the glomerulus is here, is shown in here. And then this part is the distal convoluted tubule, proximal tubule, and then glomerular. Glomerular capsule is the same as the Bowman capsule. So this is Bowman capsule, that means this is the glomerulus. Yeah, so you can see that the exit of the Bowman capsule will become the proximal convoluted tubule here. And then we have the ascending, descending limb, and then connecting duct, and then capillary. You don't see much in here, we see the cross-section. Yeah, you can see the connecting duct is relatively larger than the ascending and descending limb, because it gathers all the so-called filtrate, that means the urine from different nephron. Urine formation is divided in 3 steps: glomerular filtration, tubular reabsorption, tubular secretion. We'll go through each step one by one in details. So this is like the overview. The glomerular filtration, as the name suggested, it occurs in the glomerulus in here. So as I mentioned that the bigger afferent arterial and the smaller efferent arterial create a positive pressure inside the glomerulus capillary network, so that water, urea, glucose, amino acid, uric acid, and salt, different types of salt, they will all being so-called filtered out, or you can call it squeezed out, from the blood into the Bowman capsule, the starting point of the nephron. And then all this will travel to go to the proximal convoluted tubule, loop of Henle, and then distal convoluted tubule. And then eventually they will all join to this collecting duct. But in the proximal and the distal convoluted tubule, you have reabsorption and secretion occur. The tubular reabsorption, basically what happens is that nutrient and salt from the proximal convoluted tubule, they will come out and then reabsorbed by the blood vessels. And the secretion will be the opposite. For example, iron, and most importantly, drugs that we don't want, will be going out from the blood vessels into the tubule, the nephron tube. And then in the loop of Henle here, it is because of the positive pressure created in here, it is mainly the reabsorption, tubular reabsorption here. So we'll first talk about glomerular filtration. Glomerular filtration occurs when blood, because of the afferent arteriole is bigger, we have the positive pressure to force or to squeeze the water and small molecule into the bone capsule. Large molecule cannot leave the capillary. Large molecule, for example, protein, red blood cell, white blood cell, platelet, all these cannot leave the glomerulus or the glomerular capillary. Other stuff, I already told you, glucose, amino acid, or urea, all kinds of stuff. The previous slide that we talked about. Water, urea, glucose, amino acid, uric acid, salt, they can all go into the bone capsule. So, or we call this glomerular filtrate. These filtrate, they have all these molecules. So unfiltered molecule, I told you before, protein, red blood cell, white blood cell, like that, they leave the glomerulus through the efferent arteriole. So this table tells you the summary, but if water, nitrogenous waste like urea, uric acid, nutrients like glucose, or amino acid, salt, different kind of salt, non-filterable blood component that will exit through the efferent arteriole, blood cell, platelet, and protein. So glomerular filtration, we filter 180 liters of water per day because we have millions of nephron. So the two kidneys together can filter 180 liters of water. And so this is a table that I think is interesting. We feel two kidneys can filter 180 liter of water, and then we excrete only 1.8 liter. 1.8 liter. If you, as I told you that one ceramic coffee mug is approximately 250 milliliter. So the mass media tells us that okay, you have to drink 8 cups of water. So 8 times 250 is equal to 2,000. 2,000. That means 2 liters of water. So they suggest us to drink 2 liters of water every day. Why? Because every day we pee approximately 1.8 liter of urine. You can see that from 180 to 1.8, it is approximately 99% of the water reabsorption. And then sodium. We filter that much and then reabsorb that much, and it is also 99.5%. Remember at the beginning of the lecture I told you that you have to remember or understand this important theory is that we regulate the amount of water reabsorption by regulating the amount of sodium reabsorption. So how much sodium or how much salt reabsorb, then the water will follow. If you reabsorb the sodium, the salt back to the blood, then the water will also go to the blood by osmosis. Glucose, you have 100% absorption in healthy individual. For if we have some glucose being excreted in the urine, that means that person has what condition? Yes, that person will have diabetes. For urea, as I mentioned before, urea, they are metabolic waste by breaking down amino acid. Liver does this. Liver convert amino acid into urea. So it is a metabolic waste, nitrogenous waste. If it is nitrogenous waste, how come we still want to reabsorb the urea back to the blood? Have you thought about this? Why we want to take back the waste or garbage back to our body? We excrete some, but we absorb 44%. We reabsorb 44%. So what's the reason? Well, the main reason is that it's for reabsorption of water as well. The reason is that urea also contributes to the concentration of the blood or the osmolarity of the blood. So when it contributes to the osmolarity or the concentration of the blood, then it will help the reabsorption of water. In other words, urea helps to maintain the osmolarity of our blood. So we finished the glomerular filtration. Let's start the tubular reabsorption in the next video.